Take this away.
Projector, just in case.
Okay.
Okay, so now we're going to shift gears a little bit and talk about microbes. So Dr. Sobecky from University of Alabama is going to give her presentation on that topic.
Thank you. Prep hard is always a tough act to follow. So what I'm going to do is some bit of shock and awe with images and pictures, and then try and keep you awake after that. And thank you for the setup, because biology is king. I didn't think of that one before. I will use it now. So about 5.5 years ago, I was at Georgia Tech. I moved to the University of Alabama, and about August of '09, and then 6 months later, the spill happened. I have always done marine microbiology work. I'll show you in a second some of the work we did in the Deep Gulf about more than a decade ago now. And as the spill came on, much like what you've heard from other folks, we tried to get ahead of it and get down, because we knew that there wasn't a lot of baseline information, and particularly from a microbiology standpoint, not much. There's a lot of work, Rita Caldwell, Joe Leahy, there had been some work about microbiology here and there, and it was not done back then using modern molecular techniques that we can do now with fingerprints. All right, so that one will give you the sort of overview. So you see in the pictures, it doesn't matter where we talk in the Northern Gulf, clear, as we've heard all morning long, the impact was significant. It was extensive given the 87 days until the official well capping happened. And it's different than all the other spills you might have seen before. And many things we haven't talked about today, I know they've talked about in their seminars, is the dispersant is not, we don't know what that's done in terms of the changes chemically, all the things we know about oil spills. The dispersant adds a different layer of unknowns and a what if, right? So again, it's a experiment, as Joel said, we don't want to have, but we need to try to figure out how to sort of parameterize some of this and get an idea on how to address the changes. So I, as a microbiologist, and totally did not want to get into the terminology because we have bad enough terminology in microbiology, I use tar residues to encompass all tarballs, salpatties, SRBs, because if you're a microbiologist, SRB means sulfate-reducing bacteria, which confuses, right? So when you talk to your grad students and postdocs, they're like, which SRB are you talking about? The cultures I have in the freezer, or the stuff on the bench that we obviously have to come back to Kreberhart's lab and get because he's got it all. So what we've done is approach it from a sort of biphasic, looking at both coastal marsh sites as well as some of the beach areas, and we continue to do that work now. We're moving to projects in the Chandler Islands as part of our consortium project from Gomery. Now, one of the things to point out, and it's kind of been mentioned, but I want to throw it in as even more of a confounding topic for, I guess, discussion. We know that the Gulf of Mexico is a natural hydrocarbon seep. It's a cold seep, right? So there is input of oil and gases all the time. In addition to when we make it worse by our human-induced activities, through the petroleum industry, through transportation, exploration, and then the type of catastrophic spills that we've seen out with the Deepwater Horizon, where we've had a loss of life. So we did work in the Gulf with Mandy Joy and Joe Montoya about a decade or so ago now, where we used submersibles to go to sites that you've seen with Chuck Fisher's work and a number of different deep sea sites. These are about 1,900 meters, I guess, deep. And when you land the sub down there, it's hard probably to see with this, but you'll see in these areas where you see tube worm bushes, these are microbial mats, you'll see streams of bubbles coming up, both gas and oil. So this is going to, now, it's constant, volume-wise, very difficult, there's the numbers hit around, but is it clearly not what we see with Deepwater Horizon, but there is this input that's occurring in the Gulf. And that helps for microbial priming, and I'll show you in a minute why some of the oil degradation rates and the loss of oil has helped from a biological standpoint, because the system was primed genetically to see this material, see the carbon. Not so for Valdez, right, where that was a very pristine environment. It was cold versus here where it's warmer. So there's a very, very very different responses from a microbial standpoint. We use drills to get materials, and when you would do that, again, you see a lot of release of gas and oil. So it's there. It's not all over the seafloor, but you'll see these inputs. Then, as I said, we've got the situation with the spill. Tar balls, tar patties, SRBs, clearly there's a lot that we didn't realize in a very short period of time how the physicality of the oil changes as it's coming onto shore. So we've done a number of different approaches. We've done using these collected tar material, or SRBs. We've been doing the microbiology, and I'll show you those. And we've also been doing coastal work as well in biobattery. All right, so let's do it from a two-prong approach. So first, and I think this might get to some of the concerns about how long the oil, particularly the buried oil, because this, understanding the rates of how this material might turn over biologically is going to be key. Understanding what the components are that ultimately become available for microorganisms. And so I wanted to sort of take a moment and kind of go over the process from a microbiological standpoint. All right, so whatever SRB tar material you want to put in here is your starting point. If you're going to focus on the microbial activities, you've got a couple of choices from a microbial evolutionary standpoint. Microbes are primed to use oxygen. That's called aerobic metabolism. And they also can use, a number of them can also do what's referred to as anaerobic metabolism. They just simply use different, either oxygen, sulfate, or nitrate in their chewing up or eating of the carbon, which is coming out of your tar material. Now, what pH, temperature, less so for marine systems, the addition of adding nitrogen and phosphorus help to speed this rate of microbial metabolism. What you would love to have, which will not happen in a spill of the nature of this type, is that whatever your carbon source or your oil right here started as, ultimately it would completely go away through the microbial activity and ultimately form CO2, carbon dioxide, and water. We know that doesn't happen. Petroleum is very complex, lots of different constituents. In the case of the Deepwater Horizon, the sweet Louisiana crude, short chain alkanes, not as much polyaromatic hydrocarbons as we've been talking about. So each one of these spills, because of the nature of these different constituents, is going to have some impact on what to expect as an end product when the microbes metabolize the carbon. As I think Joel mentioned, microbes, just like the physical nature of it, might actually make more toxic daughter products. It's not that they make more toxic daughter products, it's simply that they're taking away all those easier, chewier, more labile material, degrading that first, and then what you get left behind is the far more recalcitrant, very, very hard to degrade material left behind. I mean, you think I see like the chrycine in different compounds. Okay, so with that, one thing to point out is in the water column, where you have greater oxygen penetration, rates tend to be faster. So you saw the things coming out of some of the studies with Harry Hazen's group in the water column near to the oil head. You have aerobic metabolism driving some of the activity. When you get closer to the shore, marshes, the beaches, and you'll probably hear from Marcus, you will have slower rates of aerobic metabolism. It's going to drop the rate. It'll happen. The anaerobic microbes will still profit. They'll still continue to eat it, but it's going to take a lot longer. It's going to hang around a lot longer. All right, so we've done, one of our first studies was in Pointed Pines near biobattery. We saw oil, and the only time we visually saw oil was in July 2010, and that's what looked very moose-like, came in along with the plant rack in the marsh. Didn't see it prior to that, and then never visually saw it after July 2010. We also collected the SRBs that we've been hearing about. We did the samples early on through this time period you see here through 2010. We also then went back down, got more samples after Hurricane Ivy. The sole person knowing or asking, are these very different microbial fingerprints now on these different materials. All right, so if you could turn, if it's possible to turn live, you might see this better. So what we've done is use variable pressure scanning EM to look at the tar, the SRBs, if you will, and what you'll see is if you were to look at, this is a surface scan of the tar SRBs, and you'll see intact microbial cells, as you would expect. They're picking up sand, they're in the water column where microorganisms are, bacteria. You're going to expect to see it, and we did. When you go into the interior, if you try to pull apart essentially this very, very, very sticky material, just like Prabhahar talks about, you'll see also within these interior zones of the tar balls, also you'll see colonization of a lot of microorganisms. So we knew they were there. We saw intact bacterial cells, but the question now is, who are they, and are they living? What are they doing? So just a quick reference to methodology, because I think this is where, in the last decade, advances in DNA-based sequencing and all the things you hear about in the common press have made a tremendous difference on understanding and how to profile microorganisms in these systems, which you could never have seen before, right? Very difficult to do. Now, we can go in and take any material, and we can recover nucleic acids. Now, the key thing to point out here is that if you do it right, and this is basically trying to get DNA out of your engine block, so it takes a little bit of time, but if you do it right, you can extract both DNA and RNA. Now, there will be no clicker quiz, but do not forget this, because DNA hangs around a long time. If you are a Jurassic Park fan, right, that's a little excessive, but you will have DNA in material. Does it mean that there is a live dinosaur there? No. Right? So that's the key thing to first remember is DNA means somebody's here or been here. Right? Doesn't necessarily mean you have intact cells. Right? So that's why we did that SEM image to make sure that we indeed had intact cells. Now, RNA is the way to think of RNA is that's the material that's going to take it, will be transcribed from DNA that makes the proteins, that makes the enzymes to keep the cells all running, right? So if you separate out your material correctly and get DNA and RNA, one method of investigation is to look at the DNA and say, aha, I know who's in my system. All right, and I'll show you that in a minute. If I get the DNA out, then I can say, who's active, who's living, what genes are they making or expressing, what proteins are being produced. So two very different approaches, but they're significant in how you interpret some of the data. You can also do traditional microbiology methodology, which I started 20 years ago with isolating bacteria in cultures in the lab. We still do all that. You have to do all that in order to have some working bacterial strains. All right, so let's take a look. There's a lot, so only look at the colors because names in this case really are not going to be as important. So we've been doing a longitudinal study for over 5 years now, started back in June of 2010. So what I want to point out first is that in June of 2010, even though we've never visually seen oil in June of 2010, we detected when we were pulling the salt marsh cores. Through July, we were still detecting in 2010, and then subsequent and all the way since then, it is below detection. Now, our cores are only 10 centimeters, 10, 11 centimeters in depth. They're not as deep. Doesn't mean it's not there just because you don't detect it, but we don't see it. It's at below detection levels in our system since July and September of 2010. What I want you to also look at is look at the color changes. So early on, when oil is present in the marsh system, you get a very different microbial fingerprint. Then as you go over time, it starts to look similar, right? Meaning that there are different organisms showing up now by DNA, right, than subsequent into the marsh, or into the following years. Now, if you just focus on one group, this is a group that called Bacteroides. Name's not important, but what's important is what you'll see is that when you go back and look at the different patterns, those groups, whether they're either in yellow or that dark blue, you'll see that some of the groups increase in abundance, and we think in response to the oil coming into the system. Some disappear or drop down in abundance. So there is a dynamic, a very rapid change in microbial fingerprints, as you would expect, because this carbon's coming in, this oil's coming in, now it's time they have something to metabolize. If you take all the data, aggregate it all together, which depresses my grad student, which is, it's all one slide of her dissertation, what you have is the microbial fingerprints from June and July are very different of 2010 relative to all the subsequent samplings. Now, what does that mean? It means that there's something different. Does it mean that it has recovered back to what it was previous to the spill? We don't know that data. We don't have those materials. But clearly there was a response by the microbes in the early stages of the spill coming into these marshes in Alabama. Okay, so to wrap it up, I'm going to do this and make this as painless as possible, because there's way more microbiology than most folks would like to hear. Okay, here's the samples from the tar material, the tar balls and the SRBs that my grad student at Kayla, Illinois has done. She got DNA, as I said, out of the tar material, which was, on its own accord, is amazing. And again, she did just like what Suja has done in the previous Mars data. She showed all the different groups, and she could get an abundance ratio. Again, just DNA, right? And what she found was that in some of the tar balls, we would see organisms that would predominate. This is a blue square, or blue pie, that have known capabilities of degrading dispersant components, all right? These are associated with the SRBs. She also found that the microbes, as you would hope, also would have genes for the degradation of some of the hydrocarbon constituents, like alkanes. She also found that anaerobic organisms, if you remember that chart of the metabolism, were also present on these tar materials. So all looking good. She can go in and get the genes. I won't bore you on all that, but what she can show you is she can check off the genes that are present in her DNA sequences for these different metabolic pathways that would suggest that if the organisms were active, they would be degrading, say, some of these components from the PAHs. Now, one of the things that has been a question, I think, out in the literature for a while now is, do these SRBs serve as vectors or fomites? If you work in a hospital setting, you know fomites are anything that can pass organisms back and forth, right? Do they carry microbes in to the shoreline and expose and cause health risks? And the answer is you really don't know that right now, because here's what it is known, is that if you go in and do the fingerprints, yes, we do detect organisms that would appear to be opportunistic human pathogens. You'll hear Vibrio, we know that's out in the water column. Certain times of year, the water warms up, Volnificus, and other species show up that cause problems. But here, again, here's something to keep in mind and to think about as we go forward and continue doing these studies is, many of those studies were using, again, just that DNA-based fingerprinting method. That says that the genetic material is there, but is the organism intact and actually growing? Is it active and is able to cause disease? Just because you detect an organism doesn't mean that it's capable of causing the disease. So you've got to look at all of the other factors, like the virulence genes, all the things that we can talk about later. So just because you detect it doesn't necessarily mean that it's posing a risk. That has to be followed up in a more substantive way. All right, so just to wrap up, everything we put together, again, using these modern molecular techniques in a very robust and rigorous way, I think you can begin to get an idea of what the microorganisms are doing in these systems, and ultimately being able to tell you what kind of services they're bringing to the ecosystem. And that obviously is key, no matter what, whether we continue looking forward, having, whether or not the oil stays in some of these systems or not, or how are we going to address it. So with that, I will stop, hopefully on time, and thank a whole slew of folks. With our funding agencies, of course, as Provost mentioned, you can't do it without the funding agencies' help. All of the grad students, Suja, Kayla, and postdocs, Melanie Beasley and Rob Martinez, have been instrumental in getting materials. and really overcoming a lot of the technology issues that we've had to deal with in dealing with some of these molecular-based studies. And with that, I will thank you.